everybody, Justin here. Welcome back to another Whiteboard Wednesday. And today we're looking at the mystery of multiple sclerosis. Can we do better? This is a very mystifying, confusing disease for a lot of people. I'm going to try to get through this as best I can, but there's a lot of information. And I'm mostly going to focus on infections and how that has an initiating role and also can perpetuate the disease as well, all right? But a lot of people are stuck with conventional medicine, obviously, and their treatment options, which are helpful, but can we do better actually and get to the root problem? But first we need to identify what those root problems are, okay? So as you may know, right, the immune system is attacking the central nervous system, which can include the brain, the spinal cord, and even the optic nerves. But it's demonstrated by degeneration of the myelin sheaths. So the myelin sheaths are like this coating that surrounds your neurons and it provides insulation for them so that they can transmit nerve impulses or electrical signals from one neuron to the next. So therefore we find demyelination of the neurons. We have axon injury, which is another part of the neuron, and that causes inflammatory responses and neurodegeneration. Now there are basically three different stages. We have the primary progressive MS stage or the secondary stage that occurs when it's progressive enough to cause deterioration for over 10, 15 years or so. And those are gradual worsening progressions essentially. But 85% of people with MS have relapsing remitting, which is basically an oscillation between recurring symptoms and a temporary reprieve of symptoms. And it goes back and forth that way. Research has shown more or less that, at least according to population studies, that the life expectancy is decreased by about 5 to 10 years compared to the general population. And it's largely not due to MS, but due to comorbidities or other conditions and issues that are going along with MS. So how is MS diagnosed, right? Well, there is no one reliable test. It's a collection of various data points, kind of like how it is for lupus, for example. But largely based on clinical judgment. We have the MRI that's typically done to check for brain or spinal cord lesions. We can check for IgG antibodies in your cerebral spinal fluid, or they'll even check for illegal clonal immunoglobulin bands. But none of this is specific for MS, okay? The imaging methods, though, like the MRI, is helpful, but it's not a strong predictor for MS progression. Otherwise, there's spinal tap, and checking for clinical symptoms of muscle pain, fatigue, blurry vision, poor motor patterns, cognitive impairment, numbness in the face, extremities, things like that. Outside of that, we have the alternative methods that might be helpful, right? We can actually check for antibodies against certain parts of the brain that can be indicative of MS. So one of the ones that you may not have heard of that I like is from Cyrix Labs, and it's array number 5. Now, array number 5 is basically a systemic autoimmune reactivity screen that looks at antibodies specific to, well, in this case, neuronal regions of the brain, right? However, it will also check for antibodies against various organs as well. But within the neuronal region, they're looking at myelin basic protein. They're looking at synapsin, and they are looking at, I can't really pronounce this one, but it's ganglioside. Don't ask, but those are the 3 that they're looking at. So if you check out array number 5 through the website, or through Cyrix's website, that's how you can find more about this and your doctor can order it. Pathophysiology, so how does MS develop? Well, there are a couple theories here. We don't really have a concrete answer yet, according to the research, but one of them is that there's brain damage, like a TBI or traumatic brain injury that's driven by inflammation, right, where your neurons or glial cells are really active and they breed inflammation. It could start off out as an inflammatory problem in your brain, but a neurodegenerative process occurs after that, years down the road. Okay, and then lastly, MS could be a neurodegenerative disease by itself where inflammation then comes as a secondary response. All of these, though, could occur simultaneously, but the evidence from what I've seen suggests that it's probably a combination of the first two, right? Where there's an inflammatory process of demyelination, right, in the early stages, and it transforms into a neurodegenerative process, which is just aggravated by these activated microglial cells that are in your brain. So obviously the immune system is involved here, but these are the guys that are purported to be significantly involved in the progression of MS. So we have B cells, they make your antibodies, they make pro-inflammatory cytokines, they act as antigen-presenting cells, and they are essentially picking up viruses and bacteria and other things and presenting it to the other immune cells and say, hey, we got this problem here, and they start communicating that way. We have your cytotoxic cells, and they release pro-inflammatory cytokines like TNF alpha, and that causes a lot of damage to your neurons. We have microglial activation. So these are, this and astrocytes are part of your glial cells, along with ependymal cells and oligodendrocytes. So they're part of a family of glial cells. But the point of the matter is that in microglial activation, you're releasing a lot of interleukin 1, interleukin 6, TNF alpha, interferon gamma, again, things that are causing a lot of destruction, a lot of free radicals, reactive nitrogen species, things like that, that are going to cause death to your neurons, and cause a lot of damage and disrupt the myelin, okay? And then astrocyte, actually one more thing though about microglial is that they are responsible for actually maintaining the integrity of the blood-brain barrier, right? So we have that gut lining that we want to have, but we also have that brain lining, right? So if there's a breach in that blood-brain barrier, that's where things go awry. So the microglial cells interact with your T cells and mast cells. So when they get activated, the microglial cells that is, that's where things start to go haywire, right? And they make a lot of these free radicals and they damage the myelin that way, okay? They also release this thing called substance P, which can activate your mast cells too, and that can also cause disruption to the blood-brain barrier, and once that's broken, that's where things can enter, and that's when you can have a lot of, havoc go on. Now, astrocyte activation is just another way of explaining this, in the sense that they're going to release a lot of, pro-inflammatory cytokines as well. They're going to attract more white blood cells, and if they're activated, they can damage the blood-brain barrier too, and initiate the B cells and get them moving again. So everything's kind of connected, that's what's most important here. All right, so now we're gonna look at the crux of this discussion, which is on infections. So what's important to understand is that there is no evidence that suggests there's one single infectious agent that causes MS. In fact, infections probably, so far, are not really the cause of MS, but in fact, they're part of a larger group of underlying factors that contribute to the disease. But if nothing else, they can accelerate the disease process. I think infections are missed, and that's a problem because that's probably why we think we can't do much else except take drugs. But we know that infections are involved to some degree in some people. And the studies pinpoint that even during childhood or early adult life, right, if you incur infections and environmental factors, that can set the stage for MS. And like I said, it kind of contributes to this this antigenic load, this bucket, right, where things over time fill up, and then suddenly it spills, overflows, and you have these symptoms of MS. So infections can actually create relapses after you've had MS because of the strong inflammatory response that is occurring. So the known infections that we have identified in the literature, not all of them, this is not comprehensive, but from what I found, Epstein-Barr is probably the number one. We have HHV6, okay? We have HERV, which is human endogenous retrovirus. We have chlamydia, hepatitis B, Acinetobacter. We have Staphylococcus aureus, Clostridia, Borrelia, Lyme disease, and Akkermansia. So let's talk about a few of these infections here. Epstein-Barr is the first one I'll mention because this one is strongly connected to MS. However, the evidence is not definitive. We have evidence that there are Epstein-Barr proteins that are expressed in the brains of people with MS though. But there are some theories as to what's going on here, but basically the first one would be the cross-reactivity thing, which is essentially where your antibodies and your T cells, they are seeing this virus and they cross-react and they start attacking that virus and they attack your myelin antigens, that are surrounding your neurons, and because they look the same. Okay, so that could happen. We have this heat shock protein theory where basically they are secreting a heat shock protein that attracts your T cells, and that can go on to attack your oligodendrocytes, which are in your brain, and they're going to cause demyelination. All right, we actually think that your antibodies, because your antibodies will be infected by Epstein-Barr. Epstein-Barr virus likes to really hang out in your B cells. They like to live there and take residence there. We think that they can cause a resistance of apoptosis, which basically means that the B cells never die, right? They are basically disallowing them from cell death, which is sometimes what we want to do. And then if that happens, the virus can then make its own proteins and make its own antibodies. Next, we have HHV6, human herpes virus 6. Epstein-Barr is HHV4, by the way. Now, this virus here causes encephalitis, roseola, but we found that it can live in T cells and it's been found in glial cells in your brain. The challenge is that HHV6 is a virus that's acquired many years before the disease develops. So it really confounds things, and we're not really sure whether or not the virus is really involved in MS. However, it may be something that reactivates and then exacerbates MS, as opposed to it being a preliminary triggering factor. Human endogenous retrovirus. So surprisingly, our genome is comprised of 8% retrovirus. We all have them in us. They get passed down from our mother through pregnancy, but they're usually inactive and don't cause any issues, but they can get reactivated and initiate viral transcription, and then you have a whole mess. Now, these viruses may be involved in the progression of MS, but it's not confirmed in the literature, really. Now, animal studies and in vitro studies have pointed out that Epstein-Barr, HHV6, HSV1, the flu, varicella, that causes chickenpox, right? These can reactivate the retroviruses. And the theory is that if you have something like this going on, they produce an envelope protein, which is basically like triggering a massive T cell immune response. And if that happens, we have a disruption of the blood-brain barrier potentially, neuroinflammation, and impairment of remyelination. So that's what the studies have pointed out there. Chlamydia is one that does not have a lot of cogent evidence, but we know that the virus likes to live in your macrophages and your glial cells, and that can cause the release of pro-inflammatory cytokines that can thus damage your neurons. So Akkermansia is a bacteria, Akkermansia municiphilis, if I'm saying it right. And so this one can cause immunological changes that promote MS. We actually have found people with MS to have a lot of, obviously, gut dysbiosis, where they have Akkermansia, they have overgrowth of this, what else? Ruminococcus, Methanopervibacter, and these are all associated with pro-inflammatory pathways. So there is potential for these sort of strains to be involved in MS as well. And lastly, Borrelia burgdorferi. This is the one that is indicated in Lyme disease, and the evidence shows that there's this cross-recognition between Borrelia and myelin basic protein. So your antibodies are seeing this bacteria, they see this OSPA, which is the protein that it's finding, and it sees your myelin, and it's like, hey, that looks identical to that bacteria. And that's why that cross-reactivity happens. So this can actually happen not only in your brain, but the spinal cord and even the dorsal root ganglia. So again, this is important to understand, guys, because you might want to rule Lyme disease out if you have MS. I've actually had clients who have MS and Lyme disease. Do I think that Lyme is involved? I do. So it might be worth considering, that's all. All right, so I'm going to quickly run through the different mechanisms behind how infections contribute to MS, or autoimmunity really as a whole. I've talked about this in a previous Whiteboard Wednesday, but I'll run through this quickly. So if you want to nerd out with me for a minute, here's your chance. All right, so we talked about molecular mimicry. That's obviously where infections carry similar amino acid sequences that are similar to your own proteins in your body, causing immune cells to cross-react and get confused, like in your neurons, right? So pathogens, right, they have a protein that looks, again, like your own tissue. And pathogens do this to, honestly, I think it's intelligent for them. It's to avoid immune detection. So they get sucked in by these antigen-presenting cells, like your macrophages, for example, and that then triggers the T cells, and then that triggers antibody production, and then you have that whole attack going on. Now, the other thing that can happen too is that when pathogens start to damage your own cells, your own self tissue, your own self proteins start to get released, right? And so there's like this fragments of your cells going out to the open, and then they get sucked in by these antigen-presenting cells, and then they start presenting that to your immune system. So you can see how they're actually inappropriately taking your own proteins and presenting it to the rest of your immune system, and you create antibodies that way. So that's how pathogens can do that. Okay, epitope spreading. This is when a pathogen causes a cell, like a neuron, to go through lysis. So it essentially bursts and it's dead, right? And it releases all of its contents. And so the antigen-presenting cells pick that up, they activate the T cells to say, hey, take this, and they activate the B cells, they make antibodies, so they get antibodies against your own tissue. Bystander effect is when your own tissue becomes a bystander in the immune attack. So essentially, if there's a war going on and there are people nearby and they get wounded, well, that's a problem, right? So what the process is that these antigen-presenting cells, they take the bacteria, for example, they present it to the T cell binds to it, but in doing so, these cells release pro-inflammatory cytokines and mediators, right, to communicate to other immune cells, right? But they don't have any control. So they kind of destroy everything in the vicinity because they just start releasing cytokines everywhere. And in the process, they damage your own tissue. And guess what? It causes the release of your own self-antigens or proteins that get sucked up by the antigen-presenting cells again, kind of like an epitope spreading. So that's kind of the issue right there, is that you're getting a lot of collateral damage from this effect. And then super antigen. So bacteria can invade a cell, like we talked about, and they can make their own proteins known as super antigens. And then they get to the cell membrane, the T cells bind to it, and then they say, that looks a lot like my own tissue over there, I'm gonna go attack that. Basically, you're inducing the molecular mimicry phenomenon that we talked about before. So that's the fourth thing that can happen here with the infection. So if we're looking at this here, this is the blood-brain barrier. This is like your blood vessel space, and this is your brain, okay? And you have astrocytes here, and you have microglial cells here, right? Part of the glial cell family. Now, if you have something like a bacteria, right? A virus, and you have then your antigen-presenting cell, like a macrophage or something, and it's right here, and it starts picking up this virus or bacteria. And it presents part of that protein to, well, it puts it on its cell membrane, and then it attracts this T cell. Okay, oop, wrong color. It attracts this T cell. So this T cell, when it becomes activated, right, it can then do two things. It will start to release pro-inflammatory cytokines, interleukin-1, interleukin-6, TNF-alpha, I think. And so that can activate this microglial cell and create a lot of inflammation that way, right? And start damaging nearby tissue, your neurons, and so forth, or itself for that matter. And the T cell here can actually then start to move its way down the blood-brain barrier, right? And what will happen is that it can start to squeeze inside, get inside there, and start interacting with this microglial cell. And that just basically is perpetuating the whole, communication of pro-inflammatory cytokine release. Okay, and that's really what can happen too with these infections, which is why it's so important to understand this. And then lastly, I'll touch on parasites or helminths. So here's the thing, not all parasites are created equal. I'm not talking about parasites being an issue or a trigger for MS. I'm actually talking about them being a useful tool that maybe we want to explore. Here's the thing, maybe you've heard of the hygiene hypothesis. That's the notion that people who grow up in a rural environment, they're exposed to dirt, they play in the dirt, they're in less sanitary conditions, third world countries like that, they have less allergies and autoimmune disease. We do know that from the research, compared to somebody who lives in the city. It doesn't have that exposure. And here's the crazy thing. These helminths are smart. And what's happening is that they are basically controlling the immune system and trying to suppress it so that it doesn't attack it. But you get that overall benefit, and that's what we've seen. So they induce T-reg cells, which basically put a damper on this whole process, which is good. You want that to happen because T-reg cells are dysfunctional in autoimmune disease, really. So they induce that, and they induce these anti-inflammatory cytokines, interleukin 10 and TGF-beta, which is really great. So the evidence really overall has shown that there is autoimmune downregulation in people with MS who have been inoculated with helminths or parasites. So it's an idea that you can explore. I've had clients who use it and it's worked very well, outside of MS even. But again, it's not a very common sort of intervention, but one that you can explore if you find a functional med doctor, somebody who's competent in that arena. Okay, that includes this Whiteboard Wednesday, but bear in mind that there are probably very likely other antigens, other factors, food things, mold, heavy metals, mercury, environmental toxins, things like that, that are contributing to the disease. Infections are just one element that I wanted to focus on because I think it's one missed part that does not get enough attention that could be contributing to the onset and the perpetuation of MS. So I would definitely consider ruling out infections like Lyme disease if you actually want to get to the bottom of it and heal and reverse your MS. So that's it. Hope you enjoyed it. I'll catch you next time.